the conventional drugs, they usually attack one mechanism, right? But cancer often escapes by activating another mechanism or another pathway. Cancer therapy must solve a lot of different problems, right? So it needs to select the correct patients, reaching the relevant tumor cells, penetrating the tumor in an efficient way, overcoming resistance, controlling toxicity. There's a lot of problems that we need to solve. But
in 2019, you won a European Research Council grant, starting grant to the tune of about 1.5 million euros. You are not just an academic, you are also an entrepreneur. What is a scientific failure that has taught you something important?
Well, we learn much more from failure than from success. That's for sure. And it's the failure creates the need.
Professor Conde, thank you for joining us on Research and Science Diaries, and welcome.
Thank you so much.
We are very excited to have you on the program. And how are you doing today? And how is the weather in Portugal?
Oh, it's great. Yeah, it's really sunny and warm, so it's very nice.
It makes a good time to have a conversation on nanoparticles.
Exactly.
So you are a professor of precision medicine and oncogenomics.
Yes.
At the Nova Medical School, Nova University of Lisbon, that's in Portugal. You are also the group leader of Cancer Non-Medicine Laboratory at the Comprehensive Health Research Center, Nova Medical School. You obtained your PhD in nanobiotechnology. That was back in 2013. That was at the Nova University in Lisbon. Together, I think it was a joint PhD with the University of Zaragoza. Then after that, you were also a Marie Curie research fellow, postdoctoral research fellow, and you did your postdoctoral work in so many top-ranking universities across the globe, in MIT, Harvard, as well as the Queen Mary's University here in London. You are also a co-founder and shareholder of Therapeutic. That is a biotech company that is focused on developing targeted therapies for glioblastoma multiforme. This is just a brief of your CV. Maybe you want to tell us more about your academic and professional background.
No, I think you didn't miss anything. I think more importantly is that we have been developing a lot of new technologies and new products that I hope it can be used in the future for patients, for cancer patients. Mainly, we work mainly on cancer, although we have other disease models, but mainly on cancer. And that's it. I think you didn't miss anything.
Yeah, okay. Talking about your research journey, when did you realize that you wanted to explore biology, medicine, or discovery?
Well, it was very soon, actually. I cannot point to an exact moment, but because it was something that I developed gradually, I was always fascinating with how living systems work and everything related with nature and human body. I was always attracted to understanding what actually cannot be seen directly. So I always liked this idea of the cells, how the cells work, and how they divide and how they share their information. So, I knew, the only thing I knew is that it started very soon. I was probably around 9 to 10 years old.
Yeah.
When I was in my 5th grade. I knew I wanted to do something related with biology, or with science, or with biomedical science. So I knew from that time. I didn't know if it was something more related with the lab or something more related with something more with like animal biology or plant biology, things like this. So, yeah.
Yes, but talking about the age that you started being curious into biology, I also noticed that you are also very you do a lot of science outreach, science
Yeah.
communication with kids, in the primary schools and early secondary schools. I think all through, I think you have so much engaged back in the community. What does this mean to you?
It means a lot. Actually, I always go. Every time I'm invited to go to a school, I always go to give a class for younger students. I think they are the best actually because they make a lot of questions. They don't have fear. Most of the adults, they tend to be fear. They have fear of doing dumb questions or things like this. So,
Yeah,
and they are special because of that, because they still didn't lose the the fear of asking without
yeah, without
no,
hesitating.
yeah, exactly,
That is
exactly,
that is totally true. So
exactly.
you started in biology and molecular genetics before moving deeply into nanobiotechnology. We will talk more about nanobiotechnology in the couple of minutes. But what attracted you to the intersection between engineering and medicine?
Well, I think that the biology allow us to understand a lot the disease, what's happening, right? But the
Yeah,
engineering part allow us to intervene in this biology, right? So uh, actually, during my early work in molecular genetics, also I could identify genes and pathways that were involved in actually in breast cancer. It's when I started. But there was a lot of questions that remain at the time with, so how do we manipulate these genes inside our body?
mm,
And so nanotechnology provides this bridge. I think it allows us to design a lot of materials that can protect, for example, DNA or RNA or other drugs, and recognize specific cells, for example, or respond to different biological signals and release the treatment
yeah.
at a control location and time. So this is the beauty of using this intersection between engineering and medicine. And so I was extremely attractive to this because I think it can be transformative.
Indeed. But also your scientific journey took you from Portugal to some of the world's leading universities around the world. I just mentioned MIT, Harvard, and Queen's Mary University here in London. What did those experiences change the way you think about science?
Well, my experience at MIT was the best experience I had, the best professional from a professional point of view, actually.
Yeah,
It taught me a lot how to approach my scientific questions from a problem point of view and how to solve these problems. I also learned a lot about to think about translation from the beginning of the project. Uh, because we can have a very good technology, but it's not automatically a useful medicine, right? So
yeah, yeah,
we
yeah.
must consider the patients, the clinical procedures, the manufacturing, the reproducibility, the safety problems at the early stages. And I think I've learned this a lot, and I started to think about these things when I was at MIT, because it's their mindset. It's completely different from the other institutions where I was.
Usually working from the bench and then taking it to the clinic, usually I think it comes with a lot of limitations and a lot of failures rather than looking from a patient and then bringing and say, okay, how can I engineer something from the lab that I can solve this particular
Exactly.
problem? I
Exactly.
totally, it makes, yeah. When you entered the field of nanomedicine, what was the big scientific questions that you were trying to answer?
Well, I always think, and I think this also moves me today, which the question, it's always how can we move beyond nanoparticles as passive drug delivery systems, so and
Yeah.
create systems that are capable of interacting in a smarter way with our biological processes. So well, I'm always fascinating with questions like, can a nanoparticle, for example, recognize a particular tumor state, or protect a specific therapeutic cargo, or enter a specific cell and release that cargo only when specific conditions are present. So these are, I think it's the major scientific question that always move us and move what we do in the lab. So that's the goal is not only to deliver any drug. I think that's the thing. Yeah.
Yeah. So talking about nanoparticles, let's break it down. Let's come back to first principles. So maybe you would like to really explain or define what nanoparticles is.
Yeah, nanoparticles are like these very tiny structures, like they measure tens to a few hundred nanometers. They're very small. They are sometimes often comparable in the size of viruses.
Mhm.
And their small size allow them to interact a lot with the cells and with biological molecules and membranes in ways that conventional medicines cannot.
Mhm.
And so this is the beauty of using nanoparticles. I would describe them as like a sort of combination of a protected delivery vehicle, like, or a
Mhm.
sensor,
Mhm.
and the control release system. So they can carry a drug or genetic material and protect it, and while delivering it to the cells that we want. So this is
Yeah, your previous statement you just mentioned about active delivery and passive delivery or passive targeting. Can you maybe explain a bit what is the difference between active targeting and passive targeting when it comes to nanoparticles?
yeah. Where in the passive target we use the biological barriers and the administration systems and the tissues in order to deliver without any active moiety in the surface of those particles. For the active targeting, we use usually targeting moieties like antibodies or peptides
Mhm.
or any other ligands in order to bind to specific receptors in the cells. So we are kind of forcing those nanoparticles to interact with that kind of cells or with that kind of tissue, for example. And with the passive, we just use the regular barriers that we have in terms of administration, and in terms of diffusion through the tissues and release into the tissues, as well as the fate of these nanoparticles
Yeah.
in the metabolization of the particles from with the several organs. Yeah.
For nanoparticles, there are different types. We have those that are classified as organic and inorganic. Um, how do you choose which one to use in a particular application?
Well, we work with both in the
Mhm.
lab. For example, the organic nanoparticles are the most used are lipid nanoparticles or polymeric or micelles. So
Mhm.
we work with these three types, and we also work with organic scaffolds like hydrogels, for example.
Yeah.
And we also work with inorganic ones like metal ones, especially the gold, iron oxide, and also the metal organic frameworks, the MOFs. And so we use both of these systems,
Mhm.
and I think that the material must be selected according to the biological problem, not because one class is usually more used than the others. It's also dependent a lot on how we can what are the modifications that we need to do in the nanoparticles, and how
Mhm.
can we do it with those kind of particles. And this is a really also one way to select them. Yeah.
There are a couple of delivery systems that are used in biology for to be able to deliver drugs to disease tissues. What can nanoparticles do today that the normal conventional drugs cannot do?
Oh, they can, for example, solubilize drugs that otherwise would be very difficult to administer. This is something. They can protect the therapeutic molecules. For example, they can protect RNA from degradation. This is something that we already found it also. They can alter how long a specific drug remains in circulation. They can also reduce exposure to healthy tissues, for example, and enable a local and sustained release of the cargo. So these, I think, that are the main things that conventional drugs cannot do it.
Yeah. So one of the major challenges in cancer therapy is that cancer cells are not just abnormal cells. They are constantly changing and have developed a sophisticated defense mechanism to resist drugs. How do nanoparticles help us deal with this complexity when it comes to cancer biology?
Especially with heterogeneity also, right? And drug resistance. I think that the
Yeah.
conventional drugs, they usually attack one mechanism, right? But cancer often escapes by activating another mechanism or another pathway, or changing the cellular state, or by receiving signals from the surrounding tissue, for example. And so nanoparticles can help by delivering combinations of therapeutics to the same location, or by targeting different cellular populations and releasing treatments, for example, in a sequential way. And this is something that the conventional drugs cannot do it. And so they can carry several types of therapeutics that can be gene therapies, for example, that can silence resistance pathways. Drug resistance is something that we also work a lot. And so they work on this silencing resistance pathways rather than just increasing the dose of a specific chemotherapy. So I think these are the major advantages. Yeah.
Yeah. So talking about heterogeneity in cancer biology, in 2019, you won a European Research Council starting grant to the tune of about 1.5 million euros. This was aimed at building a genetic barcode to profile breast cancer heterogeneity. Can you maybe tell us more about this project?
Yeah.
Maybe by starting by telling, maybe explain, but please try to explain to us what is genetic biocoding.
Well, what we wanted to study on this project was to combine responsive hydrogels with also with different types of nanoparticles that carry specific microRNAs or genetic circuits like plasmids that could transcribe specific microRNAs. In order to profile heterogeneity and therapeutic response in different breast cancer subtypes. So we actually analyzed several patient samples between the different subtypes of breast cancer, like luminal A, luminal B, triple negative breast cancer, and we compared with the 2D and 3D models of these specific cells, and explored also how the genetic profiling of these cells in the different systems, 2D, 3D, and in patient sample tumors, how they respond to a specific therapeutic, and in this case was a specific microRNAs. Our idea was to exactly know why they respond differently, because heterogeneity is crucial to understand, because two cells from the same tumor can respond very differently to the same treatment. So this was the and also led us to the other ERC that we just won 1 month ago, which was to develop a system, like a microfluidic system that could analyze rapidly how the cells respond to different treatments within the different subtypes of breast cancer. Yeah.
Yeah. So can you maybe give us a bit of some of the outcome of this project? I mean, in terms of results.
Oh, we are actually the main study was a very big study. We published several other studies during the course of the project, but the main study we are still going to publish it now, where we compared all these and how these nanoparticles um, could respond and how the cells could respond to these different nanoparticles. So we are still, this is still ongoing, and it will be out soon. Yeah.
Were you able to really pick out some indicators or some targets or certain things that happen in the course of therapy that gives rise to maybe some drugs responding, some cells responding, and others not responding to a particular therapy?
Yeah, we found several factors, but some of the factors that could also be influencing was, for example, the compactness of the cells in the tumor.
Mhm.
So this is something that is very interesting, and also the way they are distributed, and then they are, and the 3D architecture of the tumor cells. It's very, and it influences a lot how the drugs penetrate in the tissue and
Mhm.
how they respond to the different therapeutics. Yeah.
I mean, this can really help you know how you engineer maybe next generation nanoparticles to be able to penetrate maybe deeper into tumor cells. I guess.
Yes, for sure. I think delivery is one of the biggest problems, the delivery, but I think actually in some types of cancer are really an unsolved problem
Mhm.
in therapy. But it's not the only one. I think medicine can have an excellent, we can have actually an excellent molecular target and still fail because it does not reach the tumor at an effective concentration.
Yeah.
So this is something that we still need to work a lot in cancer therapy.
Yeah, you also have a project that is based on the idea of genetic circuits, delivering genetic circuits through nanotechnology. What are genetic circuits, and could you explain the concept of smart nanoparticles?
Yeah, these genetic circuits, like I told you, they work like these plasmids that transcribe different microRNAs that can be fused with different promoters, for example, and they can activate a therapeutic gene, for example, or remain inactive and then become active later. And so the idea of using this was to increase specificity because healthy cells are less likely to display um, this complete molecular combination. And so
Yeah.
the use of these smart nanoparticles, it's because they protect and deliver the circuit while the circuit interprets the molecular environment, right? So and so this smart behavior uh, emerges from the interaction between material design and biological design. So and the long term of this is to create therapeutic systems that respond to disease state rather than just releasing the same dose everywhere.
Yeah. Perfect. So I think this is kind of a follow up. So last week, you also won an ERC proof of concept grant to the tune of
Yeah,
150,000 euros. Congratulations on that.
yeah.
To
Thank you.
support the advancement of a project you coined Apollo BC. Could
Yeah.
you explain what Apollo BC is and what challenges this project aims to address?
Well, it's like a perfused platform for predicting therapeutic response in different breast cancer subtypes, like I told you before. So many treatments that perform well in conventional cell cultures or even in animal models, they tend to fail in patients because they are like static models, right?
Mhm.
So and do not reproduce the physical and biological environment of a human tumor, and so they lack a lot the mechanical properties, for example, which is something that we are going to explore, and how the extracellular matrix and interaction with the supporting cells, it's also important. And so we will use fresh patient tumor samples to create a perfused model, particularly for triple negative breast cancer and HER2 positive.
Yeah.
And we plan to compare responses within the platform with the patient's actual clinical responses and test several approved therapeutic options. Right. So the ultimate goal is to develop a functional sensitivity test that could help identify which treatment is most likely to work for an individual patient before
That
applying it in the clinics.
that is very futuristic and very interesting. I think it also could help really solve a lot of the challenges we face in cancer
Yeah.
therapy. So again, I'll come back to this delivery, this question, because we've seen a lot of promising drugs. Because you just mentioned also, I think the Apollo BC, it's geared towards this question that I'm going to ask now. So because we know a lot of promising drugs that work in the laboratories, the turn up don't reach um, the place in the body that we want them to reach.
Yeah.
The question is delivery the biggest unresolved problem in cancer therapy?
Yeah, yeah. Like I told you before, it is. I think it's one of the largest problems, uh, because actually, when we think about cancer therapy must solve a lot of different problems, right? So
Yeah,
it needs to select the correct patients, reaching the relevant tumor cells, penetrating the tumor in an efficient way, overcoming resistance, controlling toxicity. There's a lot of problems
sort of question. Yeah.
that we need to solve. But I think I see the delivery as part of the larger precision problem. We need to know what to deliver, where to deliver, when to release it, by sparing the other
No
tissues.
matter.
And
Yeah.
we are not doing that because we are treating patients most of the time the same way, um, independently of the tumor type, of the stage, but just combining different drugs and injecting the patients intravenously. And so there are different cancer types that would benefit from a different approach.
You are not just an academic, you are also an entrepreneur. You're the co-founder of Tatex, a biotech company focused on the development of targeted therapies. Can you tell us more about Tatex and what is its focus?
Just first of all, I'm just a co-founder, one of the co-founders and a shareholder of the company. I don't have any um, active role in the company actually, because
Okay. Yeah.
I like to do things before. So I was involved in all the technology developed behind the company, and that's what I'm most proud of it. Actually, I'm most proud of it because it was something that started in the lab, and then we can see it growing and growing, and perhaps being tested in humans in very soon. Actually, the company won an EIC, European Innovation Council
Yeah.
program, which was around 14 million euros to start the clinical validation of the product that we developed. So it's like it's we develop a targeted therapy for brain tumors, actually for glioblastoma, which are great for brain tumors, and includes a hydrogel-based concept for an intracranial delivery of a specific drug that blocks a specific ion channel on glioblastoma cells.
Yeah.
And so my contribution was to develop all these hydrogel technology, because when I arrived at the lab where this was being developed, they had a huge problem because they could find this drug, but they couldn't have an efficient delivery system. And so I developed all the hydrogel technology to deliver intracranially, because the biggest problems of the brain tumors is that when you inject any drug intravenously or systemically, it doesn't reach most of the time doesn't reach the brain because it doesn't pass the blood-brain barrier.
Yeah.
And so I think this really represents, it's a great example of how academic research can be transformed into a development program with manufacturing, regulatory things, clinical structures for a new therapy.
What is the hardest part of translating a scientific discovery into a therapy that reaches patients, from your experience?
The hardest part, first of all, one of the hardest part, which it's the funding, actually. It's to try to find investors that believe in your product. Sometimes the market, it's really addicted with just, it's not well prepared for rare diseases or more rare tumors like the glioblastoma, which is not so common. Is not so common as breast or colon or lung. But the problem with these patients is that it's an unmet medical need, so there's almost no solutions for these patients. And so this is the hardest part, is to find investors that want to invest in your product. So, and another thing is also the manufacturing part, right? So you can have an exciting result, but it needs to be reproducible, manufacturable, clinical,
GMP manufactured.
So all the requirements that you need to perform the clinical studies, and all to finding, because the clinical trial will be a multicentric one with more than 10 different countries. And so this, I think, is the hardest part.
Yeah. So again, it goes again to, because scientists always talk about the valley of death between laboratory discoveries and clinical efficacy. Why do so many promising discoveries, why is it that so many promising discoveries never make it to a patient?
I think that many projects are optimized for publication rather than translation. I think this is a problem. Many rely on simplified models, or small experimental groups, or unrealistic treatment schedules, or endpoints that do not reflect the clinical practice. This is something that is really important.
That is one of the weaknesses of academia, right? Publishing, like, you want to get it published.
Yes, exactly. So but the value of that is not only one gap, right? So it's a sequence of gaps between biological validation, reproducibility, manufacturing, like we told, the regulatory part. And so I think that the successful translation requires that these issues to be considered early rather than later in the scientific project. So I think this is the most important part. And it's most of the times why many discoveries failed in the value of that.
When you look at your experience with, because you've published extensively, right? I mean, extensively. Very good high ranking.
I have colleagues that publish much more than me. So
well, if you keep comparing, you will never stop, right?
Yeah, exactly. No, I never. That's the thing. I never compared myself with the others. I always compare with myself when I was 10 years ago, 5 years ago, 2 years ago, and so.
It stands again that you've published extensively, and you are also an entrepreneur. You've assisted in the development of a compound that is now going into the clinics. What skills does a scientist need to develop when moving from laboratory-based research to maybe a biotech?
First of all, they need to be very resilient, right? So, and also, science is also a very precarious profession, I think. Um, Before people can get some permanent position, they sometimes, and I always was from scholarship to scholarship um, for many years, for more than 10 years. And so this is something they need to be resilient and not only work hard. Working hard is but it's being resilient and never give up. Give up. It's something that it's really important. But the other skills is that they must learn how to prioritize. So academic research, I think that rewards a lot exploration, while the biotech field requires decisions in a specific time, right? And funding and capital and regulation. And so a lot of important skills are some of them that most of the times you don't learn in academia or in the lab. It's the ones that are included about intellectual property strategies,
Yeah.
regulatory science, manufacturing, clinical development, team building, communication, all of those things. And that most of the times you just learn when you are creating a company or when you are working for a company. Yeah.
Has entrepreneurship changed the way you choose a research question?
No, yeah, definitely, yeah. I now think earlier about much earlier in any project about the clinical bottlenecks that we have on that specific scientific question. What is the target population? What are the existing standard of cares? Which is very important because people always tend to think that they are developing extremely new things. We are never developing extremely new. It's very rare. We are always building up on
Previous.
yes, on shoulders of many giants, right? Of
Yeah.
giants in the scientific field. And so I think this really changed the way I view the projects. But there is one thing that I think entrepreneurship cannot eliminate. It's that curiosity-driven science. It's very important that fundamental and basic science, because there is always this fight between basic science and applied science, and I think it's a nonsense thing, because many of the transformative technologies have been originated from questions without an immediate commercial application.
Yeah.
So the important change is that when we claim translational potential, we must test the technology under conditions that are generally support that claim. But they most of them they were based on curiosity driven science. So this is something that we really need to nurture a lot.
Yeah, because I wanted to also know from your perspective, from your opinion or your experiences, moving from academic to maybe a biotech company, and how do you try to balance scientific curiosity with the urgency of creating something clinically useful?
Well, I think that curiosity expands a lot our possibilities, right?
Um,
So while the development tends to narrow these possibilities.
mm,
So I think both are necessary, but they operate very differently.
indeed.
At the discovery stage, we should explore more the mechanisms, the materials, the unexpected findings. But when the program moves to the development, we need to target a specific product, define the disease, patient groups, administration routes, everything. Um, but all the scientific decisions they are made through these different stage gates. So and a company I think cannot pursue every interesting observation, of course, but the
Yeah.
academia should, and the labs should do this. And that's it, I think. But it's very important to keep in mind that the evidence and the potential value to the patients uh, must determine which ideas we should follow
Yeah.
in advance. Yeah.
So, as of now, what is the main limitations of nanoparticles when it comes to as delivery vehicles?
Well, the nanoparticles. First of all, actually, we published some of the biggest limitations in the nanomedicine field in 2 years ago. We did one of the largest public databases where we analyzed the last 20 years of published inorganic nanoparticles for preclinical research in cancer. And we found many interesting things that were actually thought about as like rocks, solid rocks in the field, and that were not. Some of them we found that researchers they poorly report
The data,
their studies, which was something that was in discussion at the time. But what we found is that many interesting things, like for example, that targeting and passive active targeting and passive targeting was the same in terms of therapeutic response. Actually, in terms of tumor size reduction, we didn't find any difference in using targeting or a passive, an active or a passive targeting. For example, people always used to tend to use PEG, which is a polymer covering the nanoparticles. And there was also not any beneficial in using that, for example. So this was very funny to actually look at the data that have been published, and we found that. So, and we also found that the AI can also help us a lot in deciphering, and because we use predictive machine learning models at that time to see which were the nanoparticles that were best in reducing the tumor size. Yeah.
But if you just mentioned something that, okay, with the retrospective study, that overview that you did, active and passive targeting didn't show any difference in terms of clinical, let's say efficacy, because it was preclinical anyway. What do you think accounted for this? Is it the interpretation of the data back then, or what do you think is the reason behind this?
It's because it's something that we have been publishing a lot in the last years, which is the administration route plays a much important role in the preclinical outcome of some of the or most of the nanoparticles than the nanoparticle itself, which is something that it's really curious. And I think it because we can have a very potent drug, but if it doesn't get into the tissue that we want, it's going to be useless.
Yeah.
And there are some biological barriers. If we administer through IV injection or through IP or through intracranial or through intranasal, that they will the accumulation and the diffusion of those nanoparticles will be very different independently of the surface of those particles. Of course, there are some factors like the size, the shape, the charge of those particles that influence this accumulation. But I don't believe they are as strong as our biological barriers in our body. And so this is something that we really need to work on it. That's why I say that we are treating patients we are always treating patients the same way, which is most of the times intravenous injection of drugs or combinations of drugs, instead of working in different ways to administer new delivery routes and things like this. Yeah.
But what is the status quo now when it comes to the innovations that have been going on in the field for the past decades? Has there been an improved passive delivery? Has there been an improved active targeting or active delivery that has changed the way the efficacy of 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 nanoparticles based drugs?
Well, I think that, uh, for example, we when we look at chemotherapy, um, I think that it worked in the last years because it still remains effective for some types of
Okay.
tumors, but not for many of them. Um, and most of the new therapies are just new combinations.
Mhm.
But I think that we need to work better on these, and also on the nanoparticles, but also on the chemotherapy, the conventional chemotherapy, in terms of the personalization. So I think this personalization will not mean necessarily that producing just a unique medicine for one individual, right? But
Mhm.
it means integrating a lot the genomic information of the patients, which is something that we don't do a lot.
Mhm.
Integrate a lot imaging, immune profiling, liquid biopsies, for example, functional testing of patient-derived samples, which is something that sometimes we don't do it, in order to select the best treatment and adapt it to the different tumor stages. This is something that we still need to do in the future. That's for sure.
And I think this is, you said this is very important, especially when it comes to cancer treatment, because again, like you mentioned previously, you need a combination of you need to be able to combine different targeting different hallmarks to be able to get rid of cancer. This brings us to the next question, because looking 20 years ahead, what will cancer treatment look like?
I think that from all the, besides all the things that I already said, I think AI and machine learning will help a lot will help us a lot to design new molecules, new nanoparticles, new combinations, new dosing schedules. Um, it will also help us to identify patterns that humans cannot easily detect. I think
Mhm.
uh, it's something that will help us a lot. But at the end, experimental biology and clinical evidence will remain the decision makers. That's for sure. But AI, I think it will be, and the future will be. I think the future of this of cancer therapy will always be algorithm assisted,
Mhm.
but biologically validated and increasingly focused on preventing resistance before it becomes clinically visible. I think this is something that's very important. Yeah.
Do you still expect a lot of chemotherapeutic drugs to still be used in the clinic?
Yeah, I think that will still exist. Like I told before, Um, I think the problem of the chemotherapy is that for many years we only had chemotherapy as the solution, right?
Yeah,
And it saved many lives.
indeed.
Um, um, but I just think that we can improve a lot the way we deliver drugs and the way we do it, the cancer therapy. Um, so I think that, like I told you, this idea of personalization is very important, and the algorithm assisted way will be very important in the future, in the next 20 years for sure.
So talking again about science, we just discussed about the previous 20 years in your review of work based on nanoparticles. Science is often presented as a series of breakthroughs, but behind every discovery are years of failed experiments as well. Nobody always talks about these failed experiments. Anyway, what is a scientific failure that has taught you something important in
Well,
your career?
we learn much more from failure than from success. That's for sure.
Yeah.
And it's the failure creates the need to develop other things or to develop other solutions. And so this is what scientific failure taught me during my scientific path. And another thing that another lesson that came from also from the nanoparticles is that they sometimes they look very well in the tissue culture room, right? And then when you go to the animal models, this is something that is very different, and that's why I'm always very skeptical when that field of and that people that say that we don't need the animal models to test our drugs and our medicines, and I'm totally against that. I think we always need the animal models because we totally lack physiology when using the even
In
3D
vitro
models
studies,
and
yeah.
and in vitro. And so I think this is something very important to me. But the negative results, when you think about the negative results, uh, um, we should not treat them as simply technical problems. Sometimes the formulation or the nanoparticle or the drug is not the main problem. Sometimes it's the biological model
Yeah.
or the therapeutic hypothesis that we need to change, and sometimes it's this, and people always tend to look at it's the drug, it's a problem
The
of the
compound,
drug,
yeah, this is true.
yeah, of the component. So yeah.
If you have a young student, let's say you have a young Joa back maybe 10, maybe at the age 10 when you grew your curiosity, or maybe later when he was 16, 17, struggling to choose a career, what would he or she study back in college? What advice would you give to them as well as early career researchers?
Well, the most important advice I would give is to learn how to ask important questions. Actually, do or ask important questions is more important than give good answers, because good answers can become outdated, right? But a
Yeah.
good question can define an entire field. And so these are the main advice. The other advice is not to limit yourself to too early in one discipline. I think the most interesting discoveries often occur like between biology, medicine, engineering, chemistry, and computation, for example. So you must learn and you must drink from all of these fields and find good mentors who are challenging you, but also that allow you to develop and give you freedom to develop different things.
Yeah.
And actually, I was very lucky, especially at the MIT. I was very lucky because my mentor, Natalie Artsy, she gave me complete freedom to develop new ideas that I brought into the lab, and it was very, very important at the time.
What is the best scientific advice that you've ever received from a mentor while growing up, while developing your career?
I think the best advice that I received was to accept that failure is part of the progression. So I think that was definitely one of the best advices I received. Yeah.
Great. After decades of studying cancer, developing so many therapies for diagnostics as well as for therapy at the molecular level, what questions do you still have about life and biology that is still very fascinating to you?
Well, actually, one of the most interesting questions for me is, and I still remain really fascinated, it's how cells carry almost the same genome, right? Because and almost because we and even between us, we differ only from 0.1% of our genome. And so, how they can carry the same genome but still adopt to very different behaviors, right? So
Yeah.
this is something that remains, and this remain these make us also the cells to remain sensitive to the treatment, and others became very invasive. Others they became like dormant. And so why this happens, and why this type of mechanism happen, and how they change, how these states, these different states, determine the disease progression. This
Yeah.
is something that really fascinates me. Yeah.
If you could engineer one nanoparticle today that could solve one major problem in human health, what could you design it to do?
That's yeah, it's a very interesting question. I think that, for example, one of the things that I would develop would be a sort of biodegradable particle that could be capable of finding, eliminating the minimal residual disease, right? So that small number of cancer cells that
Yes.
remain after apparently successful cancer therapy, right?
Yeah.
And that later cause recurrence and metastasis. This is something that some particles that could screen our body for these remaining cells would be something that I would definitely engineer in the future. Yeah.
Amazing. I don't know. We are rounding up to it. That rounds it up today. But do you have any summary or anything you want to say before we close it out?
No, I'm just perhaps more about the mentoring part. I think it's very important. And actually, I wrote an article about leading by listening, and I think it's very important. It's an advice that I'll give to my colleagues, my principal investigator colleagues, which is always listen to your students. And give them freedom to suggest new ideas, new experiments, and I think that's the most important thing, and you will find it very rewarding at the end. Yeah.
Professor Conde, thank you very much
Thank you so
for
much.
sharing your knowledge with us. And I will keep following up on your research. And good luck with the new grant and the project that is coming up. Thank you again for joining. And to you watching, thank you for joining us today on Research and Science Diaries. And keep watching, keep following, and stay tuned to the next episode on RSD. Thank you
Thank
very
you so
much.
much. Thank you so much for the invitation. Really like it. Yeah.
Yeah.
Thank you so much.